Is this your first slide? Why, what?
Oh, no, that's my first slide. Thank you.
Okay. It would have been a good first slide.
We're gonna get I mean, it is technically the first slide.
Yeah.
Oh,
Okay, I think we can get started as people slowly trickle in now. So I'll just very quickly introduce Cliff Bohm, who's a huge part of this community and has also helped the organization of the conference quite a bit, was in charge of our art program. And we're lucky enough to have him give us a tutorial for the next 90 minutes on a tool for studying evolving systems and digital brain. I'll be taking care of the Q&A, so feel free to ask questions in the chat, and I'll keep a track of them and ask them whenever there's a breaking point. And eventually we can have a more informal Q&A where we can unmute each other and start a discussion, or however Cliff sees fit. You're the master for the next 90 minutes, so take it away.
All right, super. So yeah, my name's Cliff Bohm. I'm a PhD student at Michigan State University. I've been developing this. I'm interested in general evolutionary, like theoretical evolution, and digital evolution. So I'm going to talk about MABE today. MABE is well, we'll get to it. So first, MABE. Why? What? But first, microscopes. So a few hundred years ago, there was this guy named Lee von, I don't know how to say his name, and he was able to develop some microscopes that were 100 years ahead of anything else. And he saw things that basically nobody else could see. And he became an honorary member of the Royal Society. And he never told anyone how to make his microscopes. And no one was able to replicate his work for 100 years. And I always wonder, where would we be in terms of biology if he had shared his technology and we hadn't had to wait another 100 years before we could look at microbes. Each one of his microscopes were one-off. They were handmade by him, and each one was its own thing. If you want to start a new lab today, you're not going to hire this guy. You're going to go buy one of these. The reason you're going to buy one of these is because you know how it works. It's not going to be finicky. It's got replaceable parts. Once somebody's trained on how to use this, you could go to another one. It's probably going to be pretty similar. So what I'm talking about is handmade versus mass-produced and homemade versus outsourced. And what I'm talking about is modularity and standardization. So questions are, can we take advantage of modularity and standardization in digital evolution research? Are there standard elements, and how can modules help? So now I'm going to actually tell you what MABE is. So what is MABE? It stands for Modular Agent-Based Evolver. So MABE was first envisioned as a Markov brain evolution system. I'm going to talk more about Markov brains a little bit later, so I'm not going to talk about them now. And I put on a caveat here, at least that's what I was hired to build. So the ability to swap out environments so that we could test Markov brains in different environments was critical to the initial design concept. But as I started working on it, I realized with a little bit more work, I could allow MABE to support more than just Markov brains. And I could allow it to support different kinds of genomes and selection methods. So MABE is a tool for designed to evolve and study populations of digital agents, particularly with evolving digital brains. So what's the MABE design philosophy? Simple is better, first and foremost. We identify concepts that are common across all, or at least most, digital evolution systems. We provide support for these common concepts. We don't support features partially. So I'm going to stress this point. If there's something in the design or there's concept and we can't come up with a way to fully support that feature, the design choice is to not support the feature at all. The concern is that if we provide a partial solution, that partial solution will get in the way of people's research. So if we put something in place and that becomes a hindrance to how you need to do it to conduct your research, then the tool doesn't work for you. And so what we'd rather do is just provide no support and stay out of your way, let you take care of it. Also, really important, simple is better. So what do I mean by a common concept? File I/O, data management, parameters, configuration, population management, lineage tracking, a concept of genomes, some sort of heritable, mutable data, brains or controllers or solvers, depending on what community you're part of, evaluation methods or worlds, selection methods. These are all sort of common concepts in digital evolution research. I'm going to broadly break these down into two categories. I'm going to say that this set, these sorts of things are fixed. So once you solve file I/O, once you solve a parameter or a configuration system, you don't have to write it again. It's going to work. You could basically move that to any experiment. Genomes and brains, your evaluation methods, your selection method, these are more fluid. So if you're conducting an experiment and that experiment's about artificial neural networks, well, then you need to be using artificial neural networks. It's not going to work for you if you have to use a Markov brain. But you may have other questions. You may be interested in different mutation scheme, and that may be very specific to the type of genome that you're using. And so these elements are the elements that we basically say these elements need to exist, but we can't fully define them. So there's this very loose concept of an interface, and basically we say the concepts that can be fixed across all or at least most experiments are core elements, and the fluid concepts are modules. The interface is a theoretical distinction between the core elements and the modules. MABE's generality is only possible because it doesn't make any attempt to define the modules aside from how they interact with the rest of MABE. So what happens inside a module is none of my concern. All I care about is how that module is going to interact with MABE. It's really up to whoever is designing that module to fill it out to determine what's going to happen on the inside. So MABE then is the common elements plus communication. That's what it's really providing. So we can look at this from like a very high level. Here's sort of a MABE functional overview. We've got genomes. They're pretty much what you think. It's heritable, mutable data. You've got a brain. It's pretty much what you think. It's in our case, it's an input-output device. It takes inputs. It does a process. I don't know what, and then it generates outputs. We have an organism. Oh, he's cute. We have a world, which is going to evaluate the organism. We have an optimizer. The optimizer is the thing that selects organisms and determines which ones are going to go on to the next generation. There's a caveat here in that we don't always need an optimizer. And then there's an archivist. And the archivist is the part of the software that determines what data we're going to save, how often, and basically when we're done. When have we saved enough data? So that's all the job of the archivist. There's this last node here, which is the group, and a group is, it's a combination of a population of organisms plus the optimization method and the archive method. So the modules here are the archivist, the optimizer, the genome, the brain, and the world. The organism is just a bag that holds genomes and brains, and a group is just a collection of organisms. So in addition to the modules, MABE provides a set of utilities. These are things like a file manager, which keeps track of which files are being used during a session, data map, which is a container that allows data to move between modules, the parameters system, which allows users to, allows the world and genome and brain developers to add parameters to their system, and then some other helper functions that are just generally useful. If we look at MABE from a 10,000 foot view, this is kind of what it looks like. MABE is this piece of code that sits in the middle of the modules, and the modules are all designed externally, but more realistically, I think that this is a better view. So MABE is really, it's a very little thing. The core of MABE is quite a tiny thing, and the code that's necessary to implement the different brains and genomes and worlds, that's where the bulk of the code lies. MABE really, it's just this connective tissue that allows these modules to talk to each other. Another way we can look at MABE is as this dialematic. And the idea with the dialematic is you just plug in a world, you plug in a genome. So I'm going to say I'm going to run with this berry world. That's kind of a harvesting world. I'm going to run with a circular genome. I'm going to run with Markov brains. This simple optimizer, which is like a basic tournament optimizer. And then my archiving method is going to be this LODWAP, which stands for line of descent. And so you basically dial in your modules and that creates your experiment. If you need a module that doesn't exist, like you wanted to run, you know, here I don't have a, well, I can't think of a brain type that I don't have in this list, but that's okay. Let's say that you're working on a type of brain. Oh, I don't have ANN in that list. Great. So if you want to run with an ANN, you would have to build the ANN and then add it to the system, but you wouldn't have to build any of the other parts. Those are all, if they already exist. So the idea is once a module exists, and I'm going to repeat this idea a few times, but once a module exists, it can be used interchangeably with other modules. Generally speaking, we use a standard evolution loop. You create an initial population, evaluate the population and world, use scores from the world to select parents, generate a new population, apply mutations, rinse and repeat until you either run out of time or you get the result you want. With a caveat that we don't always follow that rule. So we can write a world such that the population is passed to the world, and then the world handles reproduction itself. And this is for cases where you want reproduction to occur immediately when an agent collects enough resource. In that case, the world takes care of it. It still uses, and then it doesn't use an optimizer, but it still uses all the other parts. It uses the archivist and everything else. This is a good stop. I'm going to go in. What I'm going to do next is I'm going to switch to a different slide presentation, and I'm going to talk about, I'm going to do a practical example showing Markov brains in berry world to give you an idea of like a low level view of what's going on. But before I do that, this is a good point to stop and ask questions if there are questions.
At this point, nothing in the chat. People feel free to unmute if you have any questions.
Okay, I will go on then. Okay. So let's talk about Markov brains for a minute. So what I'm showing here is two movies. The left is showing some published work from Olson et al. Showing evolution of swarming behavior. The white agents in this case, the little dots, are prey, and the red dot is a predator, and the predator can pick off prey, but it has this predator confusion effect where if it has too many prey in its field of view, it has trouble picking them out. And so they did this work to show that predator confusion can result in swarming behavior among prey. This thing on the right is showing a patch harvesting task. It's basically a Markov brain that's just driving this agent, trying to capture as much food as it can in a limited amount of time. So Markov brains have also been used to study, I have another, no I don't, I closed it. That was silly. Markov brains have been used to study a bunch of different evolving systems. So they've been used for classification tasks. They've been used for value judgment and perception, a bunch of behavioral tasks. So they have a wide range of applicability for research. So technically, a Markov brain is comprised of three components. It has nodes, which are numeric values, gates, which are input-output devices. They take inputs, they process the values, and then they generate outputs. And they have wires, and the wires connect the nodes and the gates. What is the gate? In this case, what I mean by a gate is a processing element which converts some number of input values into output values. So for example, here are two examples of gates. An OR gate takes two inputs and generates the OR of the two inputs. So it takes two binary inputs, it generates a binary output. This threshold 5 gate takes an input of an integer and then compares that integer to 5. If it's less than 5, it outputs 0. If it's greater than or equal to 5, it outputs 5. So a gate can be any logic. So this is what a schematic for Markov brain looks like. We have input nodes at the top in blue, gates in the middle, and then output nodes in yellow at the bottom. And the data flows from the input nodes through the gates to the output nodes. On each update of a Markov brain, each of the gates is fired in parallel. Technically, they're fired in serial, but since the outputs are ORed, it doesn't matter. But all the gates are fired. All of the data moves from the blue to the yellow. So we basically load whatever the world state is into the blue boxes, run the brain, update all the gates, and then read the output from the yellow boxes. So here's Berry World. Berry World is a world that we've used to study speciation, evolution of intelligence, sexual selection, and other topics. The rules for Berry World is it's a grid world where every position on the grid has a berry, and the berry is either blue or red. The organism has a limited number of actions in its lifetime, and on each turn, it can either turn left, turn right, move forward, eat, or take no action. If the agent eats and then it moves forward and basically leaves an empty spot, the spot will be filled randomly with either a red or a blue food. So the agent receives a score, and it's the number of berries eaten minus the number of times it switches food type times some switch cost. So if it eats a blueberry, it gets a point. If it eats another blueberry, it gets a point. If it eats a red berry, it gets a point for eating that red berry, but it loses the switch cost. So the idea is it should try to minimize, the agent should try to minimize how often they switch which type of food they eat. The agents in this case have 4 inputs and 3 outputs. And all of the inputs and outputs in this case are binary. They don't need to be, they just happen to be in this world. And so the inputs are, is the thing I'm standing on red, or is the thing I'm standing on blue? Is the thing in front of me red, or is the thing in front of me blue? So the agent here is standing on blue, so the red down is off, the blue down is on, and it's looking at red, so the red forward is on and the blue forward is off. The agent has 3 outputs, and these are move 1, move 2, and eat. The way that they work is sort of like tank driving sticks. So if move 1 is on, the agent will turn left. If move 2 is on, the agent will turn right. If 1 and 2 are both on, the agent will move forward. And if this third output, the eat output, is on, it overrides the moves and the agent will eat whatever berry it's standing on. So we can consider a very simple Markov brain. It only has a single gate. There are 4 inputs. They flow into the gate. Gate does whatever it does, and then it generates these 3 outputs. So this is a 4 in, 3 out gate. In this case, okay, and then we're gonna, if we had our agent standing on a blue berry looking at a red berry, the inputs would be 0, 1, 1, 0. It just so happens that the logic in this gate generates 1, 0, 0. So the logic is it would do a move 1, which would be a turn left. So I'm going to replace this stuff on the right with a logic table. So this logic table shows is all the possible 4 value inputs that this agent, that basically can come into this gate with the associated outputs. And we see that currently the input is 0, 1, 1, 0. So the output's going to be 1, 0, 0. So it's going to turn left. Now, before I go on, I want to note that a bunch of these inputs are not possible. It is always going to be the case that the agent may eat the food it's standing on. So it may be the case that that the down sensors will both be off, but the front sensor will always have a food. So this line is not possible. It'll never see it. Also this line, the berry in front of them cannot be both blue and red. So I'm going to remove all the lines that are impossible. The way to think of these lines is they're still going to evolve. They're going to generate values. It's just they're not under selection because they're never going to see them at any point in history. So what we're left with is these 6 lines. So let's drop the agent in the wall. It's standing on blue looking at red, and so the output's going to be that it's going to turn left. So it turns left. Now it's standing on blue looking at blue. The output is it should eat. Standing on nothing looking at blue says it should move forward. Again, we're on blue looking at blue, so we eat, move forward. Standing on blue looking at red means turn left, turn left, turn left. So now it's standing on blue looking at blue, moves, eats, moves forward. And now we've got a problem because we're standing on blue looking at red, and we're going to do that forever. We're stuck. So this logic, it gets you so far, but eventually you're going to get stuck. Essentially, this logic is if the thing in front matches the thing down, eat. And if the thing in front doesn't match the thing down, turn left. And if the current location is empty, move forward. So this agent will eat whatever food it starts on. There, you may have noticed that there were 3 lines that we didn't use. That's these 3 lines. These 3 lines are what happens if the agent starts on red. So this would be, I'm on red looking at blue, I should turn left. I'm on red looking at red, I should eat. I'm on nothing looking at red, I should move forward. This behavior we've actually seen evolve in an agent. It's not a great behavior because the agent is purely reactionary. That's sort of the downfall of this behavior. So we can do better, right? We can add a concept of a hidden node. And hidden nodes can do a few things in a Markov brain. One is we can have a hidden node that sits between 2 gates, and this allows for multi-step logic. We can also have a gate that writes to a hidden node and then exclusively reads from that hidden node. And this would act as a memory for that gate. This makes this gate sort of like a stateful gate now. If I take these hidden nodes and I move them down here and then also place them up here, you can see that my outputs would still all go to this bottom line, and my inputs would all come from this top line. I'll label this top line T, I'll label this bottom line T plus 1, and then I'll add a dotted line here to indicate that these values need to be copied every time I'm going to run this brain. And oh, hey, look, we now have a recurrent network. So if you have a recurrent ANN, it's the same trick. We just add some extra outputs and then make those recurrent inputs. I'm being sneaky, and I'll refer to what I just did later. So you're probably wondering now, hopefully, how do we build a Markov brain? We can build a Markov brain in a lot of ways. Generally, we build them from genomes. And by a genome, I mean a lot of numbers, generally something in the range of 5,000 to 20,000 values in the range of 0 to 255. So let's just look at a piece of this genome. The first thing we do is we define a set of start codons. In this case, a 43 followed by a 212 is going to be the start codon that defines a deterministic gate. We're now going to read a set of values following that start codon that are going to give us the properties to the gate. The first thing we need to know is the number of inputs and number of outputs. I have determined up front that I want my gates to have between 1 and 4 inputs and 1 and 4 outputs. That's an arbitrary choice. But in order to get a number between 1 and 4, I just take the value in the genome, mod it by 4, and add 1. I do the same thing for the number of outputs. Now I need to actually know the the input wiring and the output wiring. So I'm going to read the next 4 values. And I'm going to use similar trick. In this case, I'm just assuming that this brain had 10 inputs. So I'm modding by 10. I don't use the 4th value. I'm doing that because there were only 3 inputs. If a mutation changed this to 4 inputs, I don't want to frame shift. So I'm going to leave that 4th value unread. I'm going to read the next 4 values to use as outputs. And I'm also going to leave 2 of those unread for the same reason. And then from the next 64 values, I'm going to read only a few of them because this is only a 3 in, 2 out gate. And I'm going to just, if these are even, then they're going to be odd. And if they're, if they're even, they're going to be 0. And if they're odd, they're going to be 1. And that's going to give me my logic table. So I can look at it this way. So here is the gate from the start codon, number of inputs, number of outputs, the input wiring, the output wiring, and then the rest of these values make up the logic table. Here's the definition of the gate. Here's the logic table. So that's how we build a Markov brain. Obviously, we can have more than one gate. Every time we find a start codon, we would build a gate. We could also have a different start codon, something other than 43212, and that could define a different type of gate. So how do we evolve the brains? I showed you the evolution loop before, so I pulled it out of this slide. But when it comes to mutations, we can do a point mutation, pick a spot, change its value, copy mutation, pick a segment of the genome, make a copy, deletion, pick a segment and delete it, crossover, sexual reproduction, take 2 genomes, splice them, put them together. This method of using start codons is particularly applicable or useful in combination with sexual reproduction because the genes are relatively short to the entire genome, frame shifts aren't really a big deal. So to sort of wrap up the Markov brain sort of model, we take a genome, we convert that genome into a brain, we evaluate that brain in a world. We use a selection method to select parents, generate new genomes, rinse, repeat. So we just keep doing this. So back to MABE. So once a module, be it a brain or a world, whatever exists, it can be used in combination with other modules. This simplifies conducting follow-up research because it simplifies combining existing experiments. So if you see somebody's work and you're like, wow, I really want to continue what they've done, you can pick up MABE and then you can just extend it by adding the module you need. So it's been built in such a way that it will be extendable. You won't have to hack something into it. Additionally, if you see 2 pieces of work and go, wow, and they both happen to have been done in MABE, you may be able to just pick up the world that may be in one paper and the brain that may have been in another and just combine them and then see what happens. There's another idea that falls out of this design though, and it didn't become apparent to me until I had been working with MABE for a while, and that's this concept of substrate independence. So substrate independence asks, this is the same question, just phrased 2 different ways. To what extent can we alter a substrate in which a system is implemented and continue to drive similar results? Or does a result describe an actual phenomena, or does it simply illustrate an artifact of the particular system being employed? So as an example of substrate independence, I have these 2 visualizations from a patch harvesting task. This is the same world, same genome, same selection method, but it's different brains. So I want to stress that the code that's running the genome, the mutations on the genome, the parent selection, the agent moving in the world, that's all identical. It's the same code. The only difference is when the world asks the agent, hey, here's what you see in the world, what do you want to do? It's got a different brain. And we get these 2 rather different behaviors. We get this reasonably nice back and forth plowing. And in this case, we get this sort of S-ing, we get some sort of, a lot of repeated visitations to the same location, very inefficient. And then this kind of inefficient S-ing along the edge. So this behavior was generated by a Markov brain that had neuron gates in it. Neuron gates they're spiking neurons. So they have a threshold, they have a current charge, inputs accumulate in the neuron when they exceed, the inputs accumulate in the current charge. When that exceeds the threshold, this gate can fire and deliver its charge. The threshold and the charge can be set so that they can be modulated in real time. So in theory, this thing can learn. It's unclear if it does or not. And it generated this path. If we look at the recording of the neuron activity, the brain that generated this path had 7 neurons. Some of them are activating neurons and some of them are repressed neurons. And here's their firing patterns. This is like a little snippet of the firing patterns that generated this behavior. How this firing pattern, set of firing patterns derives this behavior, I have no idea. I haven't done the analysis. I don't know that I want to. On the other hand, we've got this behavior, and this was generated using a wire brain. Wire brain is based on a cellular automata called wire world. The idea is basically imagine a cube of copper. It's kind of special copper. When you introduce a charge to this copper, it can only travel in one direction, and it has an overcharge rule. So if there are too many connections, it'll short circuit, and then it'll kill the charge. So and that allows for diodes basically, and that allows for pretty much arbitrary logic. So it ends up that this wire world was Turing complete. When I read about it, I said, well, that's cool. I need to turn that into a cube and see if I can evolve brains out of it. And you can. Here's the brain that made this behavior. If I played the animation of this brain, it just looks like a strobing mess. And I have no idea what the logic is in this brain, but it generates this behavior. So, you know, here are 2 very, very different kinds of brains. This brain was built, they were both built using, like I said, the same, evaluated in the same world, using the same types of genomes, same mutational operators, same parent selection, very different results. So I want to sort of expand this out. I'm going to get super theoretical for a minute, so I apologize. Let's look at a bunch of different kinds of digital architectures. So we could look at classic GP, we could look at artificial neural networks, Cartesian genetic programming, reservoir computing, recurrent neural nets, tangled program graph, LSTM, wire brain, Markov brains. When I see this collection of things sort of thrown at the wall like this, I kind of see this. I think that there's relations between these different kinds of digital architectures. There is clearly some evolution. There's some conceptual evolution. Some of these ideas clearly are the result of previous ideas, but some of them aren't. Some of them derived completely de novo. So I'm not saying that there's an evolutionary history here. So when I look at this phylogeny, I'm not going to say, oh, we should be able to find the historical phylogeny of these different kinds of digital architectures. But I do think that there's a relatedness. I think that there's some kind of a mapping where we could figure out clearly ANNs and RNNs have a ton in common. You know, they must. I think wire brains probably have some stuff in common, actually quite similar to RNNs. Markov brains, it's not a hard transformation to go from the definition of a Markov brain to an RNN. So we've got all these different kinds of brains, and from where I'm sitting, and correct me if I'm wrong, but it feels like the people who work on different cognitive architectures remain relatively insulated, and they don't have a good way to communicate directly or to compare directly. And so one of the goals of MABE, or one of the goals that evolved out of working on MABE, is to make it so that it can support this kind of work. So imagine if we had 100 brains, 100 different kinds of brains, and if we had 100 different worlds or tasks, whatever you want to call them. And I asked the question, if we just took every one of the brains and we tested on every one of the worlds, we just did the big chart, would there be classifiable patterns in rates of success? So would it end up that certain types of brains perform better on certain tasks? I'm sure that there would. And I can argue, I don't even have to do the experiment. I can argue at least in one case, if you ran an ANN on a set of tasks, and you ran an RNN on a set of tasks, clearly the RNN is going to perform better on tasks that require memory, right? And the ANN is going to fail entirely. It's got no memory. So, I mean, right out of the bat, having memory or not having memory would be detectable, classifiable. But the question is, are there other patterns? Are there certain types of brains that are going to be really good at spatial tasks or classification or what have you? Would we be able to derive conclusions about the brains or the worlds? If we were given a new brain and we tested this brain on all 100 worlds, would we be able to make predictions about that new brain? Would we be able to guess at internal properties of this new brain? So other substrate independent topics are things like selection methods, mutational operators, genetic encodings, problem encodings, memory models, tasks. These are all different elements where, you know, if we look at something like a memory model, I could come up with a memory model for, say, some kind of long short-term memory, and I could apply that to an ANN, but I could take the same memory model and I could apply it to a Markov brain or CGP or some other architecture. So really, like, where working on MABE has taken me is this idea of like, and this idea is really driving me to want to make MABE, to expand MABE and make it better, is that this could be a path to some sort of unifying theory for digital evolution. And what we need is we need a platform, and whether it's MABE or something else, it doesn't really matter, but we need a platform where we can directly compare different parts of these evolving systems. And I don't want to just say the digital architecture, that's one part. The selection method is another, mutation operator is another. You know, it's work I'm hoping to do. I'm gonna stop like the lecture part and go into the tutorial part. So that's why this slide is going to show up now. So this is my thank you slide at this point. If you want to see MABE code, we're going to talk about this more, but it's located at this GitHub. I want to thank Beacon at MSU and College of Natural Sciences, and especially the Hinsa Lab, which is Arne Hinsa, who is now in Sweden. He ran away from Michigan. He got out of America just in time, who provided the funding for the first 3 years that allowed me to work on MABE. So I can take, this would be a great time to take questions if there are questions.
Yeah, I'd invite anyone to unmute themselves if they have a question. I had one just because I've played a lot with agent-based models, but always in a very applied way. So I've never had to grow, like evolve behavior or evolve brains. And you sort of, you know, you just said, oh, for our brains, to build our brains, we use a genome with a lot of numbers, and those numbers are between 0 and 255. Like, is there a way to quantify, either formally or with a rule of thumb, how much complexity or diversity you can have based on genome length and maybe world complexity? Like how big does this genome need to be? Is it just big enough? Is there such a thing as too big?
Yeah, yes, more yes. These are all open questions. So when I joined the Hinsa Lab, the general feeling was that genomes should be in the length, should be in a range of 5,000 with sort of an upward bound of around 20,000. And that was only because of computation, because converting genomes, if they're 100,000 length, gets to be kind of slow. I have played around with really short genomes, length 500, length 300, and I've been able to evolve Markov brains that have a number of gates. So these are really good questions, and again, these are the sorts of questions that I want the system to be able to look at, and importantly, I want to be able to ask that question, like if we ask about genome length, or you say like how much non-coding, if your substrate is relying on copy mutations to generate new mutable information, right? So if one of the theories of Markov brains is that they evolve by copying a gate, which generates no sorry, I saw a chat and then I get distracted. Oh, right. So one of the theories of Markov brains, and this is not published. I don't believe this is published, but that a lot of the new behaviors come about because a gate gets copied. When the gate is copied, it has no change to behavior because of the way that we basically, if they're two identical gates, they're just going to output the same. They're going to generate the same output under certain conditions, which means we can get a gene duplication, and now that second gene can mutate without having to lose the original functionality. So you can get this highly functional part, it can mutate, and then you can move through space. There's a question of how much empty space do you need in order for this to become probable? Or does it become more probable if you have a smaller genome because a copy mutation is more likely to target an existing gene? But yeah, these are open questions. Now, you could imagine, and I've done this, I have a rather weak version of CGP. It's terribly unoptimized, but I have two versions. One does a direct sort of more traditional encoding. The second one actually uses start codons. And what it does is when it finds a start codon, it reads the next value, uses that to determine the operator. It reads the next two values to determine the wiring for the operands, and then it reads a double. And the double it uses to determine the position of that operator within the list. This now makes a CGP that doesn't have any problem with crossover because it's nonlinear, so frame shifts are not an issue. And it would be interesting to see how a CGP built with a genetic start codon model has similarities to a Markov brain with start codon values as opposed to the direct encoded Markov brain and the direct encoded CGP. And being able to modify just those single variables while leaving all the rest of the code intact, that's really what I'm hoping MABE is going to be used for. I should say I'm going to caveat because I meant to put it in somewhere and I didn't. MABE has probably shown up in around 20 publications at this point. It's being used by two. It will be in a PhD thesis later this year. It was in one a couple years ago. So it's getting used. The community is still pretty small. It's probably around 20 to 30 users. All right. There was
yeah, I know you saw the question on the chat. I'll just read it for people who might not be checking the chat. So Dr. Dave was asking, is there a way to include reinforcement learning within this framework?
Yes. So we are, I was going to talk about, I'll talk about more at the end, but I'm working with Charles Lafria on implementing MABE 2.0. And this is a complete from the ground rewrite. And one of the big things that we're changing is right now when worlds and brains communicate, they communicate by a vector of doubles. So whatever the input from the world is has to get packed into a vector of doubles. It goes to the brain. The brain does whatever it wants. It generates a vector of doubles and passes it back to the world. This is limiting because if you want to pass contextual data, the brain needs to know what those inputs are. And the idea is it really shouldn't need to know. So one of the big changes that we're making to MABE 2.0 is going to be that we're going to pass kind of signal packets. So it's going to be the communication between worlds and brains is going to be a payload, and that can contain complex data. So it can contain formatted data like images. It can contain structures that have multiple pieces of data, and it can contain named signals. And this is going to allow us to pass in a feedback signal versus an input signal. Specifically, we want to be able to do that so that we can support machine learning because I think that being able to integrate machine learning, evo systems to be able to integrate these things is going to be a big deal. Actually, I think it's going to be really important, and it's also like, you know, reinforcement learning, machine learning. It's some very powerful stuff, but it has its limitations, right? The last talk was talking about limitations of these systems. So being able to integrate these two systems seamlessly would be great, and being able to pull the really good implementations of these cognitive architectures, evolving cognitive architectures, and really good implementations of machine learning, and then just seamlessly put them together without having to write all of them yourself. That's going to be a big deal. All right, cool. Awesome. All right, cool. I will go on to the demo part then. So what I'm going to do now, I'm just going to, hopefully this isn't going to be too boring. I'm going to run through sort of the here's MABE intro. So the first thing is MABE is housed on GitHub. Did I stop my sharing? Huh, you guys can't see anything. All right, there we go. So MABE is housed on, at the Hensel Lab MABE. It'll be there for the foreseeable future and probably until we get MABE 2.0. It's a wiki project. People can make pull requests and report issues. Somebody just did this morning. There's a wiki, and there we go. There's a wiki. It has some introduction. Importantly, it has the installation and quick start. The installation and quick start goes through basically what dependencies you need. Jory Shasa, who's in this talk, he actually was answering a question, has done a really amazing job of putting together this quick start guide. Basically, whatever operating system you're on, it walks you through downloading. You need Python, you need CMake, you need a C++ compiler, and he walks you through the process of getting those installed. So if you go to this document and you just do what it says, just kind of, it'll take maybe a half an hour. Mostly it's just because it has to download things. You will end up down here at the end with downloading and running MABE. And so I'm going to start with the sort of downloading and running MABE. The documentation here is okay. It could be better. It should be better. If people use it and they have questions, I am always approachable. You can contact us either through the wiki or through issues, or you can email me directly. All right, so let's get to it. So I have an empty directory here. Oh, I'm going to go back over here. I'm going to go to MABE. And I'm just going to go over here and copy the URL for MABE into my clipboard. And then I'm going to do a git clone of that and pull MABE. So it's going to take a minute to pull everything down. Oh, right, I'm on a, oh, there we go. Okay, good. Like, what are you doing?
I was going to say you're on a Zoom call, so.
I know. I was on a Zoom call before, though, when I tried this, and it wasn't too bad. There we go. Okay, good. So this gives me this MABE directory. I go into MABE, and what I have in here is we're using CMake. You don't need to know anything about CMake. We have a code directory. We have a tools directory, and we have this modules.txt. There's a README. There's a license. The first thing you have to do when you download MABE is you have to run sh tools. This is really the only like arcane thing you need to do. You have to do setup.cmd. All right, and what this does is it basically copies this mbuild.exe, which is a precompiled helper tool that will build MABE. The reason we have to run it is because we don't know what your operating system is. So when you run sh tools setup it it copies the correct mbuild, the correct mbuild. In my case, because I'm on Windows, it's mbuild exe. From here, all I have to do is mbuild. And because of the joy of CMake, CMake figures out what compiler I have on my computer and looks at the CMake list files that are propagated around the project, and is going to go ahead and build my project for me, hopefully. Oh, and I don't care how long it takes, because I'm not going to let it finish. So this would take about a minute or two, but I have.
Could you just run CMake at this point?
Yes.
Doing the mbuild? Like if you just do CMake dot, would it work?
I don't, that's a question for Jory. I know that you can CD, there's a build directory that gets generated, and you can CD into the build directory and just do make.
mkdir builds and then do CMake dot dot. Yeah, it's a typical way of using CMake. Yeah. No, just wondering if you're familiar with CMake. You know.
Jory is the rock star. So this initial build, I have gone ahead and just run that. I just want to check something to make sure I'm not doing something stupid. Okay, good. Everything is good. So I just went ahead and I did the initial build. So that's done. And what it generates, it generated two directories. One is this build directory, which we'll talk about in a minute. It's build directory. And more importantly, it generated work. And if I cd into work, here's MABE.exe. If you weren't on Windows, it would just be MABE. If I just run MABE, it's going to start up and run. And what it did was it launched MABE. It said, I'm going to run test world, and I'm building a group, and it's going to have Cartesian genetic programming brains, and it's going to use circular genomes, and it's going to have 100 default organisms. And then it ran it. And when it ran, it generated a max CSV and a pop CSV. So if we look at the pop CSV, we can see that it has a number of columns. All of these count copy, count delete. These are counting the different types of mutations that occurred. The genome length is the genome length, the optimized value, and the score. In this case, is the same value. Score is generated by the world. Optimized value is the value that the optimizer, the selector, is using. It just happens to be set to score. And then it's also tracking the current update. So on update 100, the average score was 9.95. There's a helper tool, standalone tool in tools called mgraph. And what mgraph, by default, it just looks for the pop CSV, and it just plots whatever it finds there. So it's just a very quick way to see what did we get. I'm looking at my notes. So I'm going to simplify this mgraph, and I'm just going to do an mgraph, and I'm going to add data. And I just want the score av, and I want the genome length, length av. So this will just pull up those two columns. So the score goes up and then it stays pretty high, and the genome, maybe it generally increases in length, maybe it's just random. It's hard to tell. Looks pretty random to me. Okay. So, right. So now I can, to run MABE, I just ran MABE like vanilla out of the box. So if I run MABE and I add a -s, this will save settings files. So it basically says I'm just saving settings files and I'm not going to do anything else. And now if we look, we're going to see that we have settings CFG, settings organism, and setting world. Setting CFG, well, setting organism has anything relating to the organism. So this is all about the genomes and the brains. Setting world is anything to do with the world. So the task that you're having the agent evolve on. And then settings is everything else. So it's the selection method, how long we're going to run, the population size, how we're going to save data, all that stuff. So I'm going to edit the settings file. Oopsie. And I'm going to change the output method from default to LODWAP. LODWAP stands for line of descent with aggressive pruning, which is, it's basically a line of descent tracker. So now what we're going to be looking at is the organisms on line of descent. And so now if I rerun this, I'm going to add -F and I'm going to add settings. And if there are 3 settings files here, but I want all of them. So I'm just going to do settings star. And now it's going to run, and it's the exact same run, except now I have this additional LOD data and LOD organisms. I realized I forgot to do something, but I'll go ahead and plot this anyway. It'll be silly. So I'm going to plot this, except now I'm going to add -F for files, and I'm going to do LOD data.csv. Oh, and I got something wrong. Oh, files. Oh, and I got something else wrong. Ha ha ha. Right. It's no longer the average genome length because it's now only 1 because it's in the LOD. So the reason why this is a diagonal line with only 2 points is because I'm an idiot and I forgot to do something. So in my settings.cfg, in this LODWAP, I have my data sequence, which says how often I'm going to write my data, and I'm going to write my data every update rather than every 100 updates. So now when we run it, everything is going to be better. All right, great. So that's what we get. So now we're looking at a line of descent. So we're really looking at the values of one individual and the individual that ended up being on the line of descent. The score goes up and stays at 10, and genome length kind of wiggles around. Let's look at the oh, right. Okay. So if we look at the settings file again, there's a bunch of options here. I want to call attention to 2 of these: init, pop, default 100. This is setting the initial population and saying, just make me 100 default organisms. The default organism is defined by the settings. So the settings right now say, the brain settings say that it should use CGP. The genome settings say it should use circular genome. So default 100 says, make me 100 organisms with CGP brains and circular genomes. In addition, this updates here. This is how long it's going to run. I can access parameters from the command line, so I can run MABE, and then I can add a -P and say global -updates, and say I want to run it for 500 updates. And well, this is a bit boring. Actually, before I do that, let's go into the world. Settings, world. So I'm going to go in the world, and I'm going to set the number of outputs. The task that we've been evolving is test world. It's just a max 1 problem. It's just set all of your outputs to 1. So I just made it so now rather than having 10 outputs, it has to evolve 100 outputs. So you may notice it runs a little bit slower because the brain is bigger. The way that the CGP is implemented, every output gets its own CGP expression. So I'm not going to let it get to 500 because it's already at 100 fitness. I'm just going to Control-C it, and then we can plot this. And we see what we would generally expect early on. It's quite easy to find values to flip to 1, and then it starts to converge as it gets close to 100.
The question I had, is it possible to save the individuals in the run into a database or?
Yes, so both in terms of data and in terms of organism. If we look into the, I'm already saving them. So if we look at like the LOD organisms, these are the genomes of the organisms on LOD. We can also, so this is however many genes, I don't know how many genomes. Oh, this is 1 genome because it's only outputting every 1,000 updates. So if I turn this up to higher than, if I turn that sequence up to higher than 10,000, then we can output them more regularly. We can reload organisms, so we can reload this genome file, or if it's direct encoded, then this file will not contain a genome, but it'll contain whatever the serialization of the brain is. That can be reloaded and rerun. In addition, we can save, we looked at the POP and the MAX and the LOD. We can do all the same thing with snapshots. So if we need to like capture the whole population at update 10,000, we can just output that. And we can also reload that and then continue running where we left off.
Okay.
I have a question as well, a little bit going back to my previous question. When you,
Sure.
When you specify the world and the task that the agents are trying to do, how modular is everything? Can the fitness score penalize for genome length, for example?
Yes.
Okay.
Yeah, so I can show you that really quick. I wasn't going to talk about this, but I will. So this is the roulette selector. Right now we're using tournament. So right now we're using tournament. We're using this, this line right here. What this says is what we should use to determine the optimizer value is the data map average score. Data map is a local data structure on the organism, and the score was set by the world in this case. So whatever value is here has to be a value that the world actually put in the data map. But in this case, the roulette, it's basically taking 1.05 and raising it to the score, because roulette seems to work better when you have a nonlinear increases. But this function could be anything. So within this DMAV, I can also, anything that's in the data map and the genome length is in the data map, I could grab the genome length and say, hey, your optimized value will be the DMAV score, the DMAV of the score minus 1,000 minus the genome length or something, right? So, or whatever, right? So you can make these strings whatever you want.
Thanks.
Where was I? Oh, right. So we ran that. We looked at that. We looked at genomes, right? So I'm just going through my notes really quick. They sequence that. Updates. Okay, so now I want to show you guys what's more interesting. So I'm going to cd ... And in this, so I'm in the main directory again. There's code down here, and there's this other file, modules.txt. So let's dip into code for a second. So if I ls code, this is the actual MABE code. So here's the main CPP. More importantly, here's brain, here's genome, here's optimizer, and here's world. So let's do an ls of code brain. And what we see is, well, we've got an abstract brain. If you know about inheritance in C++, then you know what we're doing here. Basically, this sets up the structure of the brain. When I talked about the communication that MABE provides communication, the communication is established by this abstract brain.cpp and .h. These are all different kinds of brains that you can plug in. So here's the ANN, here's CGP, here's a Markov brain, here's a wire brain. So what we're going to do, and then the same is true for worlds. So if we look at worlds, there aren't that many worlds, but here's Berry world, there's a block catch world, there's Logic 16 world, which is sort of inspired by Logic 9 from Avita.
Pretty impressive.
Yeah.
You've got an implementation of human brain there.
So human brain is a very useful tool. So what human brain does is, at the moment that the system asks the brain for input, it prints the input to the screen and queries a human to put in.
It's like blind watchmaker.
Well, it allows you to test, basically. It allows you to test edge cases in your world. So if you're doing a navigation world and you want to know if walking off the edge of the world will cause a seg fault, that's how you do it. You plug in a human brain and then you play the game. It's also kind of insightful because you can see sometimes you build a world and then you plug human brain in and you play the world and you go, oh my God, this is really hard. I had no idea how hard this would be. I have no idea why I think this will evolve. So, that's that's sort of the structure, right? There, if you want to add it, if you wanted to build a new brain, you would go in the brains directory, and then you'd either copy one of these. There's a tool in the mbuild system that will create a template so that you can start building a new brain, but it would go in this brain directory. Same thing for the world, the same thing for the optimizers. So from this main level, though, there's also this modules.txt, this file right here. And if we look at modules.txt, this has, this basically can go and discover what brains and worlds are currently downloaded. We have an extra repository that has more modules in it. These are just the ones that ship with MABE. But I want to go ahead and turn on Berry World, and I want to turn on ANNs, and I want to turn on Markov brains. And I'm going to write this, and then I'm going to do a /mbuild again. And so now, because I've changed the options in mbuild, in that modules.txt, mbuild is now going to build me a MABE that has all those parts, except I'm going to be mean and I'm going to kill it in a minute, because Markov brains, this would take about 3 minutes to build, mostly because of Markov brains. They just have a lot of parts. So fast forward about 5 minutes, maybe 3 minutes. And we'll go into MABE, and we'll cd into work. Oh, that was just CMake finally deciding it was done. All right, so we're in full build. So this version, if we look at the modules.txt, this has test and Berry turned on and Markov and ANN. So now, because I have that, if I look at my settings, oh, I don't have anything here, so I have to make everything again. So I'm going to do ./mabe.exe-s to save. That's going to make my settings files. So now I have settings files. I'm going to go in my settings world. I'm going to set my test world. So now there's all of the settings that I'm going past are part of Berry World. It's kind of bloated. I'm going to set my number of outputs to be 100 from test world. And I'm going to go into settings organism. And we haven't been in here yet, so I'm going to talk about it real quick. I'm going to change this to Markov. So now we're running with a Markov brain, and I'm going to go down to the bottom here. This is all Markov brain settings for the most part. This is like turning on and off different types of gates in the Markov brain. But down here we have the genome description. And so in here, we've got things like the alphabet size of the genome, the type of the genome, what data type we use to represent the sites, mutation rates, mutation sizes, stuff like that. So now I can run this. Again, it's -F and then settings star to run with all the settings. And so now we're running that task that we were just running with CGP. Now we're running with a Markov brain. And likewise, we can, should have just probably typed the command. Well, whatever, we can graph it. It's not very interesting. But what's really cool, I think, is I can do this. And I know by now you're like, well, of course you can. You said you were going to. I'm going to go into settings organism. I'm going to go to this line. I'm going to change this to ANN. I'm going to try to change this to ANN. And I'm going to run this again. And now I'm running with artificial neural networks instead of Markov brain without having to do anything else. Same world, same genome, same mutational operators. So that like literally that it's that easy. All right, where am I in my, oh, that's worth talking about. Let me run this again with Markov brain. So I can point to that. So I'll change this to Markov. And I'm going to run, and I'm going to go into settings, CFG. I'm going to put this back to line of descent. And I'm going to change this to output every update. And rerun that. Okay, so now we have our LOD files again. So I'm gonna, oh, that's not gonna work. It's not gonna work. I don't know. Maybe it'll work. I'll just do it the hard way. I want this one. Nope. All right. So I'm going to run Python. I'm just going to visualize this, but I just want to show you one extra thing now. Tools, mgraph dash data, score, av, and genome, leng length, and I'm going to do star det star. Let's see if I got that right. Oh, oh right. Files, LOD, data, CSV. All right, and no, and I clearly didn't do that right. What did I do wrong? Hold on, technical difficulties, settings.cfg. Oh, I set the wrong value. You set this one. See, even I get confused. All right. So now, right. So now we've added a third column, and this is still the score. This is still the genome length. But now we have this third column, which is the number of deterministic gates. So one of the things that MABE does is each module defines that it may be adding data to the organism's data map, and that data should get output to file. So just by running with Markov brains, you get this extra data column. And we can see in this case that the Markov brains really grow. This is because we want 100 outputs from this brain. In order to generate 100 outputs, it's got to have at least, these gates can only have 4 outputs each. So it's got to have at least 25 gates in order to generate 100 outputs. And so it's trying to get as many gates as it can, and copy mutations are the way to do it, clearly, because it's just making tons of copies in this genome. So it started at genome length 5,000 and ended up at 10,000. So now I want to show you MQ. So MQ is the system that we use when we want to do batch jobs, and it allows you, it interfaces with MABE. It's a standalone Python tool, but it interfaces with MABE and allows you to set parameters. So the first thing, I'm just going to copy from tools, MQ conditions, and I'm just going to put that here. And then I'm going to go into MQ conditions, and I'm going to change some stuff. I'm going to set this up so that it's doing reps 101 to 105. I'm gonna, what the var does is it allows me to make a shorthand. So I'm gonna make a var BT, and I'm gonna associate that with the brain dash brain type. What brain dash brain type is, if we look at the settings organism, there's a category here called brain, and within that it has brain type. So now BT is going to refer to this parameter. Whoopsie. So I have my brain type, and then down here in my conditions, I'm just going to set BT equal to first Markov and then ANN. And if I now run this, Python .tools q.py, tools, mq.py, it's just giving me a status. It says I found 2 conditions, and if I run these 2 conditions, it will result in 10 jobs. I can run this with -L, which will just run it locally. And this will just take a minute. It's just going to generate, it's just going to run these 10 experiments. So while this is running, let's see if there are any questions I can answer. Is there a way to see the questions without having to stop sharing? Oh, yes, there is. Chat. There we go. Got it.
Jory has been great to answer a lot of the questions in the chat as well.
Super.
If this is a good time for a more general question, there's a question I've been asking to everyone who talks about sort of agent-based models, just because I'm curious about your own experience or anyone else in the tutorial. What's the best way to visualize some of these outputs? Like, we've been looking at sort of average per generation, but sometimes we're interested in sort of extremal events, right? Like, some people use confidence intervals. Some people just use like particular runs, unique runs. I'm curious about you. I'm
like the worst person to ask. There's a terminology that people in my lab and in the Ofria lab, they refer to them as cliff plots, because I tend to try to aggregate way too much data in a plot. I'm also very much into watching visualization. So Berry World is actually implementing an agent running around the world, and I want to watch that and I want to see what the behavior is. So I spend way too much time looking at really complex plots. I really think it depends on your question, and the concern I always have is if you look at an average without error bars, yeah, you know what the average is, but you don't know, it's hard to tell what's going on. And it's also, you may have, if you have a bimodal distribution, if you look at error bars, you don't know if you have a mess or if you have a bimodal distribution. Also, if you just look at error bars, you don't know if there are outliers. So it could be that like 90% of your agents perform poorly and 10% do great, but they're not showing up if you only look at averages. And that may be fine based on your question. So I'd say I think that's a really hard question to ask, how best to visualize. But what do other people think?
Could I follow up on that question?
Yeah, of course, please.
So you showed Berry World. Were those visualizations part of the extensibility built into MABE, or is any graphical visualization just an entirely external thing?
So the idea, the overarching idea is everything in Berry World is actually just part of the module. So Berry World happens to ship with MABE. It ships with Berry World. If you turn on a visualization flag, you can like load an organism, turn on a visualization flag, and it'll output text files. And Berry World actually has, it ships with it a processing script that will create visualizations. So it's not doing it in code, but I kind of want to stress that MABE itself, core MABE, has no concept of space or location or direction or berries or any of that. It's not, because as soon as I start to define any of those concepts, I'm concerned that I'm going to start limiting what people can use it for, right? I'd rather be in a position where the user has to write some code, but they can have it do exactly what they need it to do, as opposed to trying to work with it. Like I spent some time looking at NetLogo recently, and I'm incredibly impressed by that piece of software. And I'm incredibly impressed by the ability to create interfaces. It has this amazing interface to drop visualizations and interface objects and develop code, but it's a grid of patches, and it has these turtles, and the turtles move on the patches, and there are links between turtles, and that's what you get. And so if that's not what you need, it's not really going to work for you, right? If that is what you need, it's phenomenal, but if it's not what you need, it's not going to work. What I really wanted to be able to do is support evolution of digital architectures, and then secondarily looking at how like different genomic structures and selection methods impact rates of adaptation. And so the specifics of the world, I really try to stay out of it. Hopefully that's I sort of sidestepped the question, but hopefully that's a reasonable answer.
Yeah, but maybe I have a follow-up to my follow-up then.
Sure.
So what I first thought I heard as the general description of this is that you have MABE, and then I would write my module for the brain, my module for the world, etc., according to your interface and definition, and plug them into MABE. But maybe I should think of it as the reverse. I create my world, and MABE is a plug-in into it, which handles certain tasks for me.
I think the first way is the right way to think of it.
Oh.
So what it does is, I mean, think about it this way. You want to do some science, and let's say that you want to know how ANNs perform in a certain environment. So that if the ANN module that we have is a good enough implementation for you, you don't have to build the ANNs. They're already done. Additionally, you don't have to worry about the parameter system. You don't have to worry about file IO. You don't have to worry about lineage tracking. All that stuff's already handled, because that's stuff that's not going to change. So we could hard code all that. If your world, you're almost certainly, most people who use MABE end up having to code a world because it's a specific experimental question.
Sure.
You may end up having to code the brain because you may need the brain to behave in a certain way or have certain mutational operators. You know, I don't know. And that's why I sort of am hands off, right? But the hope is that as people build new worlds and new brains and new genomes, those get integrated into this larger library, this repository of parts that can be pulled from. And then as moving forward, when you go to an experiment, you say, oh, has someone already done the thing I need? Like maybe I don't have to write that thing. Maybe it's already there. And that's, you know, that's sort of that's the hope of this software, right? So it does sort of, in my mind, there are two important things. One is it gets you from a hypothesis to actually being able to get tangible data. It just tries to make that as short as possible. And then the other thing is, imagine that you have this question about how some ANNs are going to perform in some particular environment. Wouldn't it be nice if you have a certain hypothesis about maybe it's the structure of the ANN or something else, if you could just, you know, offhandedly grab a Markov brain, plug it into your world and see how it does? Like, wouldn't that give you some insight into whether or not the thing that you think is causing the effect is actually that thing, or if it's something else?
Yeah.
Right, and that's for free. The idea is that that's, if you're using MABE, that comes for free. I mean, you got to run the thing, but it's as easy as going to the settings file and flipping a switch. Cool. So hopefully that answers the question. I hope I'm not Bill Gatesing this too much, or sorry, Steve Jobsing this too much. I guess I'm Bill Gatesing and I'm trying to Steve Jobs it.
You lost me on that one, but we're going to talk.
Okay.
I'll give up the floor to somebody else.
All right, cool.
Yeah, well, I just want to say, to keep you on time, Cliff, you have about 4 minutes until we're technically out of time.
Sure.
Okay.
I'm going to show two visualizations really quick, and then we should have a couple of minutes for questions. So the first visual. So we just ran this. MQ, and we ran 5 replicates of an ANN brain and a Markov brain doing Berry World. And so now that I have that, I pre-typed out my command because I'm lazy. And copy that. And let's see if I did it right. Oh, of course I didn't. Oh, well, that's fine. Oh, because I'm using pop files. Sure. We'll use pop files. That's fine. Files, pop files, that means that this has to be genome length AV. So if you look at this, I just did an analysis of the directory, and you'll see that there's a C0BT, so C0 brain type Markov, C1 BT brain type ANN. So these are the 2 condition directories. If I list C0, you'll see that there are 5 replicates, and if I list C0 101, you'll see that our if I list that would be nice. you'll see that we've got LOD data, LOD organisms, max, and pop. So basically the same data that we've been generating just goes into the condition and replicate directory. So now I can run this mgraph command, and I can get it wrong. Oh, what I'm saying here is I'm doing mgraph. I'm going to slow down because I'm just trying to rush myself. I'm doing mgraph. Files is pop CSV. Conditions is all the C conditions. Data is, I want to see the genome length av and the score av. And then my rep range is 101 to 105. And I'm going to combine conditions. And so now what we see is, first of all, out of the gate, the ANNs perform on the task. This is the score. They're much better, but this makes sense. Markov brains have very few gates initially. And so gates that are outputs that are not wired are 0. So the Markov brains have to create gates in a hurry. The ANNs, 50% of their outputs are going to be 1 and 50% are going to be 0. So they're going to get a 50% score out of the gate. But the Markov brains are catching up. If we look at the genome length, well, the story is really different. The ANN genome length doesn't really change at all, and the Markov brain genome length just goes through the roof. So this is like a real time demonstration of running 2 different brains, right? This thing that I'm talking about, 2 different kinds of brains, same world, same mutation, same selection. And we get to sort of get an insight, oh yeah, maybe that gate copying thing is causing this increase in genome length. So that's kind of what I need to say. So I'm done.
Okay, we have well, we're right on time, but maybe we can go a little over and take a minute or 2 for questions.
If anyone has, I'm also really interested in the next talk. So if we want to stop now, I'm happy to do it.
I was gonna say this is a great back-to-back. So in lecture hall A, in any minute, Leo Cassin is going to be presenting on his BIBIT, which is also evolving agent-based models. Let's focus on the theory a little bit more on the visualization of it. So
yeah,
I
and
invite
please,
everyone that's here to move. But
yeah, I'm going over there. If anyone has questions, please feel free to contact me either by email or on the Slack.
okay, well, thank you so much, Cliff. This was great.
Yeah, thank you. This was good. Thanks.